Okay, so welcome to another episode of the A Level Cookbook. Today we're going to be going through AQA A Level Biology Paper 2 from June 2019. And sorry if I sound a bit nasal today, I have been a bit ill. Succession occurs in a natural ecosystem. Describe and explain how succession occurs. As a quick reminder, what happens in succession is that you have this really hostile region. So it could be like bare rock, or it could be just like dirt with nothing in it. And what happens is a species that's very, very good at surviving in harsh habitats, something that's well adapted to harsh environments, colonizes that area. And that's known as a pioneer species. Now, eventually, what happens is the pioneer species obviously do their things. They digest, they eat, they, do whatever, and they die. And when they die, microorganisms like bacteria scran them up and other things like saprobionts and stuff. Once they're scranned up, return the nutrients into this harsh environment. So when nutrients are returned into the harsh environment, that means that there's less harsh because now there's stuff to actually be used. So species that are adapted to less hostile environments are now able to colonize this area. So this keeps happening basically where they eventually die, they return nutrients, saprobionts break that, digest them down into more nutrients, and the cycle keeps going until eventually you've got this massive diversity. So you might start off with like lichen and, weird little things like that, and eventually you get little bushes, shrubs, etc., all the way till you get trees, and eventually you hit something called a climax community. That's a summary, and obviously we need to talk about the key things. So first of all, what happens is you've got colonization as a result of the pioneer species. So you've got colonization by pioneer species. So there's the first point. The next point is that they change the environment by dying or whatever, forming a soil or something like that. So they change the environment and change abiotic factors. So abiotic factors are things like temperature, soil, etc. What happens is the environment becomes suitable for different species that are adapted to less hostile conditions. And then what they do is they change the environment as well, so it's more suitable for other species. And what can actually happen as well is they make so many changes that it actually becomes less suitable for the previous species. So these new species displace the other ones. So you could also talk about that. And what this does is this increases biodiversity and you reach a climax community. I guess an example is imagine you've just got this bare rock, like you've just got rock, nothing else. So there's this stuff called lichen, which is able to survive because they can usually just they can break down like rock and stuff. So you've got lichen here. This is your pioneer species. Eventually, what happens is the lichen die, they're decomposed, and you start to get like a thin soil, full of nutrients like nitrates, and full of nutrients like nitrates, etc. And then what can happen is eventually a new species comes along. So I don't know, like some other green plant of some sort chills here, and eventually the lichen aren't really good at living there anymore, so they're off. And then what happens is these die and then they decompose, and then eventually you start to get things like, like weeds and plants and whatever, and then they die, and eventually you start to get trees. Woohoo. So that's pretty much what succession is. You can have secondary success. So this is primary succession when you're starting from the bare rock and bare whatever it could be. It could be like really bright environments where the light bleaches everything. You can have secondary succession as well. So this is where you've got land that's been cleared of plants, but the soil is still there. So imagine like we had like a imagine somebody had a gender reveal and they set the entire forest on fire or something like that. It definitely hasn't happened ever. Then what can happen is all of this gets erased, but the soil is still there. So you don't really need like the same pioneer species at the start. It's just succession starts again. So, things start growing, whatever, whatever, but you don't have to go all the way after that pioneer stage. Figure one shows the percentages of energy transferred from sunlight to a zebra in a grassland ecosystem. So we've got sunlight, 2.1% of the sunlight goes to vegetation, 12.7% of the vegetation goes to zebra, and then the zebra uses 36.1 of it on respiration, 58.2% of it feces and urine, and then 5.7% on new tissue. Use figure one to calculate the percentage of sunlight energy that would have been transferred into the feces and urine of a zebra, sorry. Give your answer to three significant figures. Okay. Oops, let me mute my phone. So what this question's testing you on is your ability to use percentages. So when you have a percent of a percent, you always times it by the percent in decimals. So, for example, the sunlight is 100%, right? Like that's the original source we're taking it from. So, 2.1% of 100% is going to be that times 0.2, sorry, 0.021, which gives you 2.1%. Hold on, let me change this around. Okay, so whenever you take a percentage of something, you times by the percent in decimals. So for example, if I've got 73 and I want to find 25% of that, I do this times 0.25, which gives us 18.25. So in this case, 2.1% of the sunlight, so we got 2.1% over 100 goes into vegetation. Of the 2.1%, 12.7% goes to the zebra, sorry. So now we've got 12.7 over 100. So timesing the original 2.7 that came from the sun by the 12.7. And then of that 12.7, 58.2 ends up in the feces and urine. So we're going to do 58.2 over 100. Now, if you do that, you should get an answer of 1.55 times 10 to the minus 3. But the question wants it in a percent. So you times that by 100 and you get 0.155. So now it says in this ecosystem, the net productivity of the vegetation is 24,525 kilojoules per meter squared per year. Use this information and figure 1 to calculate the energy stored in the new tissues of the zebra in kilojoules per meter squared per year. Okay, so the net productivity of the vegetation. So the vegetation is outputting this much per year, whatever, right? So 24,525. Of that, 12.7% is going into the zebra. So 12.7 over 100. All right. And then of this fraction that goes into the zebra, 5.7 goes into the tissue. So 5.7 over 100. You type that into your calculator, and we should get the answer, which is 177.5. Okay, so we've got sickle cell disease. SCD is a group of inherited disorders. People with sickle cell disease have sickle shaped red sickle. Oh my god! Restart. Sickle cell disease. SCD is a group of inherited disorders. People with SCD have sickle shaped. God. Restart. Sickle cell disease. SCD is a group of inherited disorders. People with SCD have sickle shaped red blood cells. A single base substitution mutation can cause one type of SCD. This mutation causes a change in the structure of the beta polypeptide chains in hemoglobin. Explain how a single base substitution causes the change in the structure of this polypeptide. Do not include details of transcription and translation in your answer. This one should be a walk in the park. Your DNA is read in triplets in codons, and each codon forms an amino acid. Amino acids form a sequence by condensation reactions to form proteins, and this is their primary sequence. What happens is hydrogen bonds and this and that, blah, 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 cause them to fold and coil into, a weird shape or whatever. And that's your secondary structure. And then what happens is you get hydrogen and ionic bonds between everything, giving it like a more specific three-dimensional structure, like so. And that is your tertiary structure. That's how all proteins form. And as well as that, if you've got multiple polypeptide chains, you've got multiple of these coming together, that's your quaternary structure. So now this question is asking, explain how a single base substitution causes a change. Well, amino acids are determined by the DNA sequence. So if I change one of these out for another, then the amino acid might change, which can then affect all the folding and coiling, etc. So we need to talk about that. So what we have is we've got a change in amino acid sequence and change in primary structure. And what this does is this changes the folding and coiling, but we need to explain why the folding and coiling happens. That happens because of hydrogen, ionic, and or disulfide bridges. So you need to talk about them changing. So you get changes in folding and coiling due to changes in hydrogen bonding or ionic bonding, whichever one you want to list out, but just I usually just say that and that's sufficient. And what does this do? This changes the tertiary structure. This is not an enzyme, so we don't need to talk about active site and all of that kind of stuff. And it even says ignore 3D. I see a lot of people just write, oh, it changes the 3D structure. That doesn't mean, it literally means nothing. The tertiary structure is the hydrogen and ionic bonding, etc., giving rise to that 3D structure. If you just say 3D structure, that means absolutely nothing. You're not explaining that you understand the different elements of a protein. Hematopoietic stem cell transplantation, HSCT, is a long term treatment for SCD. In HSCT, the patient receives stem cells from the bone marrow of a person who does not have SCD. The donor is often the patient's brother or sister, and before the treatment starts, the patient's faulty bone marrow has to be destroyed. Use this information to explain how HSCT is an effective long term treatment for SCD. SCD is where you have sickle shaped red blood cells, which are faulty and bad. So you need to remember where they all come from. So they come from stem cells in the bone marrow. So if you obliterate all the stem cells, then no SCD cells are going to be made. So you've got no defective cells, you've got no defective hemoglobin, etc., and you're starting to make healthy red blood cells instead. So that's what we're talking about here. So if you're destroying the bone marrow things, then what happens is the new bone marrow stem cells replicate and they produce no faulty red blood cells, and they produce healthy red blood cells. The question here is trying to assess your understanding on how sort of stem cells work in application. So it's not really asking you to say like, oh, it's a pluripotent, multipotent, whatever, right? It's just trying to get you to understand that if you obliterate all the stem cells this person already has, put new stem cells in, then you're not going to get faulty RBCs, you're not going to get faulty hemoglobin, I should say, sorry, and you get, healthy red blood cells. Okay, so a new long-term treatment for SCD involves the use of gene therapy. So figure 2 shows some of the stages involved in the treatment of a child with SCD. So we've got a functional gene for beta polypeptide isolated. Many copies of the beta polypeptide gene are made. Beta polypeptide is inserted into a virus. Sample of faulty stem cells removed from the child's bone marrow. The virus carries beta polypeptide gene into the stem cells, so I'm presuming it's from the faulty stem cells from the bone marrow, and genetically modified stem cells injected back into the child. Some scientists have concluded that this method of gene therapy will be a more effective long term treatment for SCD than HSCT. Use all the information provided to evaluate this conclusion. Okay, so let's think about what needs to be done here. So I need to take a patient, destroy their bone marrow, find somebody who has a good match, and then put it in. Now, the problem with that is, first of all, I mean, you're destroying bone marrow, so they're not going to have any cells for a temporary amount of time, and that includes white cells. So their immune system's going to be pretty battered. As well as that, I have to look for a donor, and if my family members are the only match I have and they say, "No, I don't wanna do it, mate," then I'm kinda screwed. So that's pretty not good, but gene therapy doesn't really need that. So that's a pretty good advantage for this. As well as that, because I'm not taking cells from anyone else, there's not really a chance of rejection happening. My body's not gonna destroy it, my immune system's not gonna recognize it as foreign. With evaluate type things, you wanna talk about fors and againsts. So the fors are pretty clear here. So, there's no destruction of bone marrow, and, donors are not required. And obviously, there's a less chance of rejection or reduced chance of rejection. However, the mark scheme says there's a maximum of 2 marks for these 3 points because it's asking you to evaluate. You need to talk about fors and against. You can't just list all fors. That's not an evaluation. That's not critiquing it as well. So we need to think about negatives. It's a new treatment. We don't know the long-term effects of it. So there's one. New treatment. Don't know long-term effects. And as well as that, we're using a virus here, so it could actually cause problems like, it could give them the flu or something like that. A student investigated the effects of indole acetic acid, IAA, on the growth of oat seedlings, which are young plants. The student removed the shoot tip from each seedling and cut out a 10 millimeter length of shoot. They placed 10 lengths of shoot into 5 petri dishes, added to each petri dish an identical volume of 5% glucose solution, added to each petri dish 40 centimeters cubed of a different concentration of IAA. They left the petri dishes at 20 centimeters. They left the petri dishes at 20 degrees C in the dark and with their lids on for 5 days. They removed the shoots after 5 days and measured them, and they determined the mean change in length of shoot at each concentration. And here are their results. Table 1 shows their results. So we've got IAA concentration added to a petri dish in parts per million. So, go 10 to the minus 5, 10 to the minus 3, 10 to the minus 1, 1, 10. So these are going up. And there's the mean change in length of shoot. So the shoot length is going up as well. So explain why the student removed the shoot tip from each seedling. One thing you need to remember is that IAA, indole acetic acid, is produced in the tip. So they need to remove that because obviously that affects the concentration, doesn't it? Don't know why there's so much space for this, but yeah, the tip produces, which would affect. Because if you think about it, they're adding IAA to this petri dish, and if you've got loads and loads of IAA in one of them and then like nothing in the other, one of them's going to grow more than the other, and that's not controlled. It's affecting your results. So you want to get rid of it because then when you test whether IAA has an impact, you know that all the IAA is coming from what you've added and not from what's already there. Okay, so now it says explain why the student added glucose solution to each petri dish. Well, think about it. What do we need glucose for, bro? What's this success? What's this testing? It's testing. This experiment that they're testing is growth. You can't grow without energy. You can't get energy without respiration. You can't get respiration. Well, I mean, you can. You can't get respiration without a derivative of glucose. I mean, you can with like proteins, amino acids, whatever. Fine. But basically, the principle here is glucose, respiration, energy, growth. So it's for respiration to release energy for growth. Whenever you mention respiration, always talk about releasing energy, not producing, because you can't produce energy. That's not possible. You need to say releasing energy every single time. I can guarantee you, either one's a mark, or they both have to be said for a mark. So please, whenever you're referencing respiration in the context of growth, or and anything, to be honest, you need to say to release energy. So we need to think about why the lids were kept on the petri dishes. Well, evaporation would happen if the lid was on, or either you'd get changes in CO2 and O2. But I don't think it's those two because obviously those you got to remember this has only been going on for a very short amount of time, and also they've controlled everything else. So if we have evaporation, what problems does that come with? Well, imagine you've got this solution with IAA. Each black dot is IAA, right? This is how much water you've got in. Got a decent amount. Now imagine I take away all the water. Now imagine I take away a load of water and the same amount of IAA is in the same spot. The concentration is way higher, which means that's going to affect the growth because organisms use concentration gradients all the time. So if the concentration's too high, what it might do is that some of it will start to move elsewhere, etc., blah, 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 which can affect growth. So to prevent evaporation, which changes IAA concentration. Or what you could also say is it changes the water potential. The water potential is hella low here, so water might start moving in from somewhere else to balance out the water potentials. All right, so it says describe and explain the results shown in table 1 and show how the results might have differed if the root had been used. So, okay, so the table shows that as they add more IAA, there's more growth roughly. So it says describe and explain. So we're going to describe first. So increasing IAA concentration. So we're going to describe first. So increasing IAA concentration, you need to say that. You can't just say increasing IAA. Leads to a greater mean change in length. You need to say what the table says. All right, so that's our describe part. You need to say what the table's talking about, not just like, oh yeah, more IAA means more growth. That means nothing. You need to see what the table says. The table says the mean change in length. So now we need to talk about why. In shoots, high concentrations of IAA stimulates cell elongation. So that's why they're growing longer, they're growing taller. The length is changing. So high concentrations of IAA stimulate cell elongation. This is what it does. Not more cells or increasing length or whatever and this and that. IAA stimulates cell elongation in shoots. However, in roots it's different. In roots what happens is it inhibits cell elongation or growth. So in roots, growth inhibited with high IAA concentration. Because it makes sense. Because if you think about it, you've got a shoot here. All right, this is your shoot. Loads and loads of IAA down here. The cells here are gonna elongate, and that's gonna bend the shoot upwards. So imagine that's where the light is. All right. In a root, however, you want the root to grow downwards. So imagine I've got a root here. We've got hella IAA here. If the IAA made it grow, then it would go like this, and we don't want that. We want it to go downwards. So what it does is it inhibits growth. So this grows faster than this. So what happens is you get this downward bend. So moving on then. The student produced the different concentrations of IAA using a stock 1 gram per decimeter cubed solution of IAA. And there you go. That's 1 gram per decimeter cubed is 1 part per thousand in distilled water. Complete table 2 with the volumes of stock IAA solution in distilled water to produce 40 centimeters cubed of 10 parts per million. So we need to do some unit conversions first. So we've been told that 1 gram per decimeter cubed equals 1 part per thousand. So to get it from part per thousand to part per million, we need to divide this by 1,000. So 1 times 10 to the minus 3. Actually, no, sorry. So 0.001 grams per decimeter cubed equals 1 part per million. They want 10 parts per million, so we need to times this by 10. So we're going to get 0.01 grams per decimeter cubed, which is 10 parts per million. Next, we're going to use this formula, Next, we're going to use this formula, C1V1 equals C2V2, right? So C1V1, C2V2. All right, so C1 is the concentration you have, which is 1 gram per decimeter cubed. V1 is how much of C1 you're going to take out. We don't know this yet. That's what we're looking for. C2 is the concentration you want, which is going to be 0.01 grams per decimeter cubed. And V2 is the volume that we want as well. So the volume we want is 40 centimeters cubed. We need to convert that to decimeters. So that's going to be 0.04 decimeters cubed. So now we can rearrange this to find out V1. So V1 is going to equal C2 V2 over C1, which is going to be 0.01 times 0.04 all over C1, which gives us 0.01, which gives us 4 times 10 to the minus 4 decimeters cubed. So then to convert that to centimeters cubed, you times by 1,000, and you get 0.4 centimeters cubed. So from this original stock sample, we want to take out 0.4 centimeters cubed. So the volume of stock solution is going to be that. But we want to make 40 centimeters cubed. So all we need to do is we just need to add the rest in water, which is 39.6 centimeters cubed. With these kinds of calculation questions, all of that work we did was just for 1 mark. If you can't do it, don't bother, just move on. It's not worth it. The complex calculation-y weird questions in biology are usually not really worth it because the amount of work you need to put in them. So scientists investigated the effect of a decrease in pH on muscle contraction. The scientists did the investigation with 4 different preparations of isolated muscle tissue. So you got A, B, C, and D. So A is mouse muscle fibers at a typical pH of mouse muscle tissue, and that's your control 1. Muscle fibers at 0.5 unit pH below typical, so it's slightly acidic. You've got rabbit at the typical pH and whatever, so that's control 2, and we've got rabbit where it's slightly more acidic as well. Okay, fine. So they measured the force of muscle contraction of the muscle fibers at 12 degrees, 22 degrees, and 32 degrees Celsius. Figure 3 shows the results the scientists obtained for B and D compared with the appropriate control. So we can see on the y-axis we have force of muscle contraction, and it's the percentage of the appropriate control. So 100% would mean that the concentrate. Sorry, oh my god. So 100% would mean that the contraction of whatever the test thing is the exact same as the control. So it's very important you make sense of that. And then you've got the temperature here. And then obviously the black dotted ones are mouse muscle fibers and the rabbits are the white dots. And you can see that as the temperature is climbing up, you start to get an increase here. And at around these temperatures, like around whatever temperature here and here, you have the same as the original. And beyond these temperatures, the force actually exceeds the original. So a student looked at the results and concluded that a decrease in pH does cause a decrease in the force of muscle contraction. Use figure 3 to evaluate this conclusion. So with evaluate questions, if you've watched any of my other videos, you always say for and against. So you need to say what the data says and how this supports what they say and what's against it and what other pitfalls are present. So, sample sizes, age, sex, gender, what did they actually test it in? Was it in, the actual organism? Was it in test tubes, etc.? Okay, so it says to use these figures to evaluate this conclusion. So this shows the results for B and D, so the test ones, compared to the appropriate control. So what we're looking for is they're saying that a decrease in pH causes a decrease in the muscle fiber contraction. Because these are a percentage of the original controls, it should be less than 100%. So we're looking at everything that's below here. Obviously, we can see that in both of them, the muscle contraction is less than 100% of the original controls when you have, at these temperatures. So it suggests that pH being lower, so a decrease in pH decreases the force of contraction. However, as the temperature climbs up, it starts to increase back to 100 and if not actually exceed it. So we need to discuss that as well. So I'm going to look for so like I said, we need to look for every point where it's below 100% because that's where it's decreased. So around, what is that? 29 degrees. Sorry, so for the mouse, it's about 29 degrees and about 27 degrees for the rabbit. So for the mouse, so the force of contraction lower than in control for mouse B. Or you could say the other thing for the rabbit as well, which is about 27. That's 1 mark. However, there's a higher force of contraction above 29 degrees C or 27 in the rabbit because you can see it's exceeded 100%. So what that means is that they're actually contracting stronger than the controls, which doesn't really say that pH is the only thing affecting it because if that were the case, changing the temperature would have no effect. But changing the temperature, as you can see, increases it. So therefore, temperature also plays a role as well. This obviously supports their thing this doesn't really support their thing. So we need to continue analyzing what's going on here. Well, they're saying that a decrease in pH does cause this, but we've only tested one pH. Like, what if it was a 0.7 units lower or 5 units lower or 10 units lower? So we've only used one pH value. It could be that, like, strangely enough, if you change it, it might actually be higher. Maybe the pH, maybe a pH of 2.5 means it contracts really well. Another thing as well is that, like, they're saying that it causes a decrease in the force of muscle contraction. Now, this is under the assumption it's in every single organism. We've only tested it in mice and rabbits. If you tested it in humans, for example, it might be different. So only tested in mice and rabbits. What else can we talk about? The other thing that happens is well, you've got temperatures as low as like what 12 degrees Celsius. The body temperature of an animal doesn't really get to that point normally, most of them, as far as I'm aware. So these results might not actually be the same in a body because you're obviously, as you can see, the temperature's climbing up. So we don't know if the like, for example, the temperature of a rabbit might be like 32 degrees, for example. So the body temperature of a mouse or a rabbit isn't exactly the same as the ones tested. So body temperature of mice and rabbit, they're not really known. So that's what I mean. You've got to say what's for and against, and whether it's been tested in, all organisms, this, that, blah, blah, blah. And obviously, what's it been tested in? Has it been tested in the organism's body itself or in isolated tissue? Now, it's told you in the question it's an isolate. Other things you could talk about is that there's no statistical tests to see if the difference is significant. You can't just say no stats test, that's no marks. You need to say the stats test is to see if the difference between these points, or why this isn't just the same as this, is significant, because this might be chance. It might be just by luck that these are different, and in reality, that you do another time and it's like that, or if you do another time, it's like this. So statistical significance means that if you do this again, they are going to stay separate. Okay, so another group of scientists suggested that a decrease in the force of muscle contraction is caused by an increase of the concentration of inorganic phosphate in muscle tissues. Their hypothesis is that an increase in concentration of inorganic phosphate prevents the release of calcium ions within muscle tissues. Explain how a decrease in the concentration of calcium ions within muscle tissues could cause a decrease in the force of muscle contraction. Okay, so I'm going to quickly recap how muscles contract just over here on the side. So you've got your myosin filament here, and you've got a globular head. So I'm going to call that the myosin filament, and that's your globular head. And you've got an actin filament here, like so. Right. Now what happens is you have tropomyosin blocking the actin-myosin binding site, like so. So the myosin globular head can't bind to the binding site, so you can't actually get any muscle contraction going. When you have an action potential from a neuron at a neuromuscular junction, the depolarization spreads into the sarcoplasmic reticulum. The sarcoplasmic reticulum releases calcium ions, so Ca2+. Those calcium ions bind to the tropomyosin like so, and then move it out the way. So it frees this spot here. So the tropomyosin's now moved out the way. The myosin can actually bind to this and form an actin-myosin cross bridge. What calcium ions also do is they activate ATP hydrolase. So ATP hydrolase breaks down ATP into ADP and PI. This gives the energy needed for the globular head to move. So the head moves and it drags the actin filament alongside it as well, like this. It's a really shit drawing, but you get the picture. Like it bends it this way, and that ATP is needed. So for all of these processes to happen, calcium is necessary. Calcium is needed to move the tropomyosin out the way and to activate the ADP and PI. And this cycle will continue happening for as long as calcium is present, or calcium ions. When muscle contractions, so when the stimulation is stopped, when those action potentials are no longer reaching the muscles, the calcium ions are taken away from the binding sites back into the sarcoplasmic reticulum, and everything gets put back into place. So it says, explain how a decrease in the concentration of calcium ions would cause a decrease in the force of contraction. Well, I've just said here, less tropomyosin moves away from the actin-myosin binding site. Therefore, you've got fewer actin-myosin cross bridges. So the myosin head cannot move and pull actin. Or you could have also said there's no ATP hydrolase activation. Those are both one mark. Okay, so in muscles, pyruvate is converted to lactate during prolonged exercise. Explain why converting pyruvate to lactate allows the continued production of ATP by anaerobic respiration. Okay, so I'm not going to do the entirety of respiration. I'm just going to do a quick summary. So you've got glucose. And then you've got glycolysis. So glycolysis happens in the cytoplasm, and you get pyruvate. Now, depending on what scenario you're in, the pyruvate goes through different things. So if you've got aerobic respiration, it goes to the link reaction, which then, does the rest. So, you've got your Krebs cycle, your oxidative phosphorylation. However, in anaerobic scenarios, what happens? So, anaerobic, you get lactic fermentation. So, what happens, or lactate fermentation, let's say, sorry. So, what happens is the pyruvate becomes lactate or lactic acid. So, what happens is reduced NAD comes along and becomes NAD. So you guys might write it as NADH or whatever. I always write reduced NAD and NAD just because it's the simplest and most reliable way, in my opinion. So the reduced NAD reduces the pyruvate. So the reduced NAD itself gets oxidized and the pyruvate gets reduced into lactate. Why is this important? Well, in glycolysis, I should have mentioned before, sorry, is that in glycolysis, one of the things you get is two NADs becoming two reduced NADs. So the two reduced NADs, this step is needed for the conversion of glucose into pyruvate, which then is converted to lactate. And you can see that this lactate regenerates NAD, which comes back here and continues the cycle. Why is this important? Well, it regenerates NAD, so glycolysis continues. So describe the role of glucagon in gluconeogenesis. Do not include in your answer details on the second messenger model of glucagon action. So glucagon has two roles. It has to raise the blood glucose concentration. So it can either do this by gluconeogenesis, so glucose, new, genesis making. So making new glucose, or glycogenolysis. So breakdown of glycogen into glucose. Okay, so both of them make glucose. As a side note, the way I remember glucagon and insulin is that insulin moves blood glucose into cells. So it's out of the blood into cells, so it lowers the blood glucose concentration. And then glucagon is the opposite. So it says do not include details in the second messenger model. So all we're going to talk about is how it leads to gluconeogenesis. So all it does is it stimulates enzymes that convert. And we need to talk about gluconeogenesis. So what does gluconeogenesis use? It uses glycerol and amino acids. So that convert amino acids or glycerol into glucose. So just as a quick recap, gluconeogenesis, glucose, sugar, new, genesis, making. It uses amino acids and glycerol to convert them into glucose. And glycogenolysis, glycogen, lysis, breakdown, breaks down glycogen into glucose. So the gene that codes for glucagon is 9.531 kilobases in length. The DNA helix makes one complete turn every 10 base pairs. Every complete turn is 3.4 nanometers in length. Use this information to calculate the length in micrometers of the gene for glucagon. Give your answer to three significant figures. So we're going to convert this into just bases. So we've got 9.531 kilobases. So to get it to bases, you times it by 1,000. So we're going to get 9531. Now, every 10 bases, you get a turn. So the number of turns is going to be this divided by 10, which is 953.1 turns. Each turn is 3.4 nanometers in length. So you're going to do 953.1 times 3.4, which gives you some answer in nanometers. If I do that, I get 3240 nanometers. So to convert from nanometers to micrometers, you need to divide by 1,000. So that's going to be 3.24. So metformin is a drug that's commonly used to treat type 2 diabetes. Metformin's ability to lower the blood glucose concentration involves a number of mechanisms, including increasing a cell's sensitivity to insulin and inhibiting adenylate cyclase. Explain how increasing a cell's sensitivity to insulin will lower the blood glucose concentration. Lots and lots of cells in the body have receptors for insulin, and they also have a type of channel protein called GLUT4. So this is going to be GLUT4, and this lets glucose enter the cell. So if you're increasing, so how it works is when insulin binds to this, more GLUT4s are present on the cell, so more glucose could move into the cell, out of the blood, and lower the blood glucose concentration. So it says, explain how increasing a cell's sensitivity to insulin lowers this. Well, if a cell is more sensitive to insulin, so there's more receptors, and the receptors are activated because insulin's binding to it, what happens is loads and loads of vesicles will move GLUT4 to the cytoplasm like this, and now you've got hella GLUT4, loads of glucose moving in, and then lower amounts in the blood. So that's what we're going to say here. So insulin binds to receptors, should say complementary receptors, increases uptake of glucose via glucose transport proteins. Right. So explain how inhibiting adenylyl cyclase may help lower the blood glucose concentration. This is talking about the second messenger model. So what happens is that glucagon binds to a receptor. So imagine you've got your cell membrane, you've got your receptor here, and this is glucagon binding to it. It activates an enzyme called adenylyl cyclase. Adenylyl cyclase converts ATP into cyclic AMP known as CAMP via adenylyl cyclase. This CAMP activates something called protein kinase A. And protein kinase A activates enzymes that break down glycogen into glucose. All right. So it says explain how. So glycogenolysis. You can see the rest of it on the screen. So it says explain how inhibiting adenylyl cyclase may help lower the blood glucose concentration. Well, you've got less ATP converted to cyclic AMP, and then obviously you've got fewer protein kinases activated, and then you get less glycogenolysis. So glucagon and adrenaline are the two things that use adenylyl cyclase, the CAMP second messenger model in A level. So in fruit flies, a gene for body color has a dominant allele for gray body, big G, and a recessive allele for black body, little g. A gene for eye color has a dominant allele for red eyes, big R, and a recessive allele for white eyes, little r, and is located on the X chromosome. Figure 4 shows the phenotypes of fruit flies over 4 generations. So what we got here, so the squares are males and the circles are females. If they are dark, they've got red eyes, and if they are white, like these white boxes, they've got white eyes. All right, so it says give the genotype of the fruit fly numbered 6 in figure 4. So we're looking at this one here. So, okay, so it's a female, so it's got to be XX. It's got red eyes, and red eyes is capital R. However, it has offspring with white eyes, so it has to be heterozygous. It can't be homozygous. So it's got to be big R, little r. It's got a gray body, but it has an offspring with a black body, so it has to be heterozygous for that too. So it's big G, little g. Give one piece of evidence for figure 4 to show that the allele for gray body color is dominant. Start. Let's say gray is recessive. All right, so it says the gray body color is dominant. If it was recessive, then these would be little g, little g, little g, little g. It can't be big G, little g, because we've just said that gray is recessive in our counterexample here. That means that all the offspring would be gray, but that's not the case because there's a black one. So 3 and 4 would produce all gray offspring if it were recessive. Explain one piece of evidence for figure 4 to show that the gene for body color is not on the X chromosome. Okay, if it were on the X chromosome, then let's think about it this way. Let's say we have X big G here and Y. So you got a gray dad, and we've got X little g, a black-bodied female. So that's X big G, X little g. So that's it's a gray female. X big G, X little g. That's another gray female. And then we've got a black male, and a black male. So if it were X-linked, it would be impossible for a gray male and a female that's black to have gray offspring. All the offspring would be black because they all have to inherit the X from the mother. So we can try looking for that on here, and if we see that, we could see right here that this fly cannot be gray under that circumstance. It could only be black. So fly 5 cannot be gray if it were on the X chromosome. Now we need to talk about why, because if it were, it would inherit the body color allele from the female, which would be black. So a heterozygous gray-bodied white-eyed female fly was crossed with a black-bodied red-eyed male fly. Complete the genetic diagram below to show all the possible genotypes and the ratio of phenotypes expected in the offspring from this cross. Question reading complete. Let me get my bearings again because I keep bloody forgetting what they are. So gray body is big G, and they're heterozygous. It's going to be big G, little g, and it is a white-eyed female. So it's a white-eyed female, so it has to be X little r, X little r. And then it's a black-bodied red-eyed male, so it's going to be little g, X big R, Y. So now the gametes they would make are big G X R, big G X R, little g X R, little g X R, little g X big R, little g Y, little g X big R, little g Y. So those are your gametes. And now we're going to draw out the cross. So this, I need more space. So it's going to move over here. So you've got, when crossing them, keep the G's and the G's together. So if you put a capital G first, capital G comes before all of them in that way. So I would be big G, little g, X, big R, X, little r. So I usually do the big ones first. And then it's going to be big G, little g, X, big R, X, little r, Y. And then we just have to count them all up into the different genotypes. So the genotypes we have are big G, little g, X, big R, X, little r. We've got little g, X, big R, X, little r. And then we've got big G, X, little r, Y. And then little g, X, little r, Y. And the phenotypes, I'm just gonna write this short as gray, red, female. We've got black, red, female. We're talking about eyes and body here, but I'm just doing it for the sake of time. Gray, white, male, and then black, white, male. And then all you've got to do is count them all up, whichever ones match a different phenotypes, and the ratio should be 1 to 1 to 1 to 1. You can count that in your own time. Just for the sake of time, I'm not going to bother doing that. Okay, so a population of fruit flies contains 64% gray-bodied flies. So I'm just going to rewrite this as big G is gray, little g is black, big R, sorry, X, big R is red, X, little r. Population of fruit flies contains 64% gray-bodied flies. Use the Hardy-Weinberg equation to calculate the percentage of flies heterozygous for gene G. So we need to, so gray can either be big G, or big G, little g. All right. So I'm going to let big G equal P and little g equal Q. All right. So that must mean that P squared plus 2PQ equals the 64%, so 0.64. Therefore, Q squared has to equal, because of the P squared plus 2PQ plus Q squared equals 1. That must mean that this Q squared is whatever 1 minus 0.64 is, which is going to be 0.36. What we can now do is we can work out what Q is by doing the square root of 0.36, which is 0.6. Is that my mental maths correct? Yeah, so 0.6. And then you know that P plus Q equals 1. So P is going to be 1 minus 0.6, which is 0.4. Now we can work out the 2PQ is the one we want because it's asking us for the heterozygous ones. So we're looking for our GG, big G, little g, which is big G here, little g this. Now, just a side note, the reason it's called 2PQ is because you can have big G, little g, and little g, big G. They're the same thing, so that's why we call it 2PQ. So coming back to this, we know that P squared plus 2PQ equals 0.64. We now know that P squared is whatever this is squared. So 0.4 squared plus 2PQ equals 0.64. So that means 2PQ, which is what we want, is going to be equal 0.64 minus 0.4 squared, which is 0.48. However, they want it in a percentage, so all we need to do is times it by 100 and we get 48%. Okay, so in photosynthesis, which chemicals are needed for the light-dependent reaction? Tick one box. So reduced NADP, ADP, PI, water, and oxygen. Again, ATP, NADP, reduced NADP is a product of it, ATP is a product of it, and CO2 is used in the Calvin cycle of the light-independent reaction. So it has to be the last option, which is NADP, ADPPI, and water, which is correct because that's used in photolysis. These two are used to make ATP, and that's reduced to make reduced NADP. So there you go. Describe what happens during photoionization in the light-dependent reaction. So what happens is chlorophyll absorbs light energy, and what this does is this excites an electron, which is then released from the chlorophyll. So just as a quick summary, you've got light shining on, chlorophyll, whatever, blah, 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 and electrons released. It moves down the electron transport chain to photosystem 1, which then more light is shone on it, and it's excited to an even higher level, and it's passed on to NADP. Sorry, it's passed on to NADP to produce reduced NADP. Because we've lost an electron here, an electron needs to be replaced. So water is split into protons, oxygen, and electrons. The electron goes here. The oxygen is released as a waste product, and the protons. So protons create a concentration gradient inside the thylakoid and the stroma. Energy from the electron moving down the electron transport chain pumps more protons into here. Protons want to move back down a concentration gradient. The only way they can do that is through a protein channel embedded called ATP synthase, which combines ADP and PI to form ATP. So a student obtained a solution of pigments from the leaves of a plant. The student used paper chromatography to separate the pigments. Figure 5 shows the chromatogram produced. So we've got the origin here. We've got E, D, C, B, A. So they say, explain why the student marked the origin using a pencil line rather than using ink. Well, it would be that the ink and leaf pigments and all that kind of stuff would mix. So it would either dissolve downwards and mix into here, or it would mix with everything in here and give you like massive blotches like that and mess up your results. Pencil lead is insoluble, so that's why. So the ink and leaf pigments would mix. Okay, so describe the method the student used to separate the pigments after the solution of pigments had been applied to the origin. Okay, so what they're asking you is basically what did they do with it once they drew everything on. So what you do is you dunk it in a solvent, but you make sure the solvent is below the origin line. You don't want it to be at the origin line because when you're starting off chromatography, they're all here. If you put it at the origin line, it's all just going to dissolve downwards like that and mess up your results. You want it to be underneath the origin line because the solvent will move up the paper like this. You also want to stop it for the same reason, roughly near the end, because if it reaches the end, then you can't really reliably measure a solvent front because it might be that it's reached the absolute top and it could have gone further had you left it for longer. So that's why we kind of just stop it there. Place solvent beneath origin line and then remove solvent before it reaches the top. Okay, so it says calculate the RF values of the pigments that can help identify each pigment. An RF value compares the distance the pigment has moved from the origin with the distance the solvent front has moved from the origin. So that's the formula there. The distance each pigment has moved is measured from the middle of each spot. Pigment A has an RF value of 0.95. Use figure 5 to calculate the RF value of pigment C. So I don't actually have a ruler, but basically they've told you that 0.95 equals the distance that the pigments move from the origin. So you're going to have to use your ruler and measure this distance from here to here. And that's, I'm going to call that distance, I'm just going to call that distance A. All right. And over the solvent front, which we don't know. So you want to rearrange this to get the solvent front. So that's going to be the solvent front equals the distance A over 0.95. Now that you have the solvent front, you can work out the RF value of C. So C's RF value would be distance C over what you just worked out, the solvent front. So the pigments in leaves are different colors. Suggest and explain the advantage of having different colored pigments in leaves. Well, okay, so just as a side note, when you look at a plant and it's green, the reason it's green is because it's absorbing all light frequencies or wavelengths except green. It's reflecting green back at you. If you absorb all light frequencies or wavelengths, you have a black object. If you absorb none, you have a white object. Wavelength determines the color of light. So based on that, plants cannot absorb all possible light or they would be black. So the reason that you can have different colored pigments in leaves is so that they can absorb more wavelengths. That would be useful. Can absorb more wavelengths of light. Okay, so the next question says, what is a DNA probe? So it's a short piece of DNA with bases that are complementary to the target gene allele or DNA. So it's a single short strand of DNA with complementary bases to the target gene. It's very important you mention that bases are complementary. Right. DNA probes are used to detect specific base sequences of DNA. The process is shown in figure 6. So a DNA sample is broken down into smaller fragments. The DNA fragments are separated by electrophoresis and transferred to a nylon membrane. The DNA fragments are treated to form single strands and DNA probes are added. DNA probes allow specific base sequences of DNA to be detected. Describe how the DNA is broken into smaller fragments. So this is where you use restriction endonucleases. So whenever we talk about enzymes doing anything in any context, always, always, always talk about the site at which it works if possible, and also the bonds broken. So this is a restriction endonuclease, and this hydrolyzes phosphodiester bonds at specific base sequences. That's the target. So, for example, lipase, the target of lipase is lipids, and it's breaking down ester bonds, and so on. The DNA on the nylon membrane is treated to form single strands. Explain why. Well, DNA probes only. So, DNA probe is like a one strand here. It doesn't work if you've got double stranded DNA. The way a DNA probe works is you've got a single strand, and if it's complementary, the DNA probe forms complementary like base pairs with it, like that. So it's so the DNA probe binds. A scientist used DNA probes and a gel electrophoresis to screen 4 volunteers for 5 different viral DNA fragments. Figure 7 shows the results the scientist obtained. The lanes numbered 2 to 5 represent the 4 volunteers. So volunteer 1, 2, 3, 4, and this is like a control ladder, I'm presuming. So gel electrophoresis separates them based on length. So we have a ladder of known lengths here so that we can match them like this. So for example, I don't know, this is so let's say this is like a couple hundred kilobases long or whatever. Then we know that this one here matches with that, so that's got to be a couple hundred kilobases long. Without this, it means absolutely nothing to just have this on its own because all we know is that 2 and 4, 5 have similar length DNAs, but we don't know similar lengths of DNA, but we don't know how much. So that's what it is. So lane 1 has DNA fragments of known sizes. And what does it do? Well, what does it allow us to do? It allows us to compare with, the test samples. It's our reference. Okay, the lengths of the viral DNA fragments were 600, 250, 535, 300, and 500. Which volunteer which volunteers had at least one of the viral DNA fragments at 250 or 535? Okay, so again, as a reminder, the heaviest ones stay towards the start, and the lightest ones go towards here. Ones, sorry, meaning like the DNA fragments. So if you have like loads and loads of bases, it's not going to move very far. So that means that because we're looking for 5 different things, this ladder's got more than fives. It's got a couple extras. So we're going to be looking at each one of these to figure them out. So this one's got to be the heaviest at 600. And then we've got 535, then we've got 500, and then we've got 300, and then 250. So now we need to identify who has 250 or 535s. So 3 has that, 3, sorry, has a 250, 4 has a 535, and 5 has a 250. So 3, 4, 5. Okay, the sundew is a small flowering plant growing in wet habitats such as bogs and marshes. The soil in bogs and marshes is acidic and has a very low concentration of some nutrients. The sundew can trap and digest insects. Describe how you would estimate the size of a population of sundews in a small marsh. Right, so you've got this big area here, and it's not feasible to, look at the entire area. So what you can do is you can divide it into a grid, and then you assign each place like a coordinate. So A, B, C, D, one, sorry, was 1, 2, 3, 4. then you can pick spots. But obviously, we don't want to put bias. You might pick a place where there's loads of mildews because it looks good, and that's bias. So you want to use a random number generator to spread out as randomly as possible, and then you just count how many are in, a quad. So in within this quad, this section. So that let's say I've counted like one, two, three, four, five, six, right in this area A1, and this let's say this is a 10 by 10 area. So that's 100. In total, we have 1,600 meters squared area, and let's say that's 100 meters squared. So what I can do is I can say there's 6 per 100 meters squared, and then I can use that to upscale to 1,600, which would be timesing it by 16. So that's what we're talking about. So you can count how many are in one square and then scale it up by doing that, because you know that the 6 in 100 meters squared. So in 1,600 meters squared, you would do that, this whole thing times 16. Now to put it into words. So you would divide the area into sections and give them coordinates. You would use a random number generator to select sections to count. Then you would count the number of organisms in the area of the section, then divide by the area of the section. Then you would multiply by the area, well, the total area, sorry, of all sections to estimate the total population. And as well as that, you'd also use a large sample size and calculate the mean number per sample. So what this point means, sorry, is back to my diagram that I've drawn here, which I've kind of messed up now. Um, I wouldn't only use A1 because that gives me 6. Maybe if I do C2, there's one, two, three, four, and maybe in B3, there's one, two, three, four, five, six, 7. So if I do loads and loads and loads of them, work out a mean, and then do that, that's better. The reason I've mentioned scaling it up is because it's asking you to estimate the size of a population of sundews in a small marsh, not just your section, the whole marsh. So now it says, suggest and explain how digesting insects helps the sundew grow in soil with very low concentration of some nutrients. So in these questions, everyone sort of says a vague idea of what's going on, but misses what specific nutrients help. In these types of questions where it's talking about nutrient cycles, you have to name a specific nutrient. So obviously, if they are able to digest insects, that means it's the digestion involves breaking stuff down. So you need to think, okay, what's something useful that's good for a plant to grow, and talk about that. So digestion breaks down proteins, which provides amino acids for protein synthesis. You need to talk about for a use. Also, you could talk about you could say, like, for example, digestion releases phosphate, which can be used in DNA and RNA, and so on. Okay, so with these passage questions, a lot of people just kind of don't bother reading it. Try and go to lines 2 to 4 and have no idea what's going on. Just have a quick skim. And when it says lines 2 to 4, don't just stick on lines 2 and 4. Read a bit around it as well. So Guillain-Barré syndrome is a rare disease in which the immune system damages the myelin sheath of neurons. Myelin sheath damage can cause a range of symptoms, for example, numbness, muscular weakness, and muscular paralysis. Sometimes neurons of the autonomic nervous system are affected, causing heart rate irregularities. Huntington's disease is a disorder where a protein called huntingtin damages the brain. Huntingtin is produced because of a dominant mutant allele. The first successful drug trial to reduce concentrations of huntingtin in the human brain involved 46 patients. The patients received the drug for 4 months. The concentration of huntingtin was reduced in all the participants. The drug was injected at the base of the spine into the cerebrospinal fluid, bathing the brain and spinal cord. The drug contains single-stranded DNA molecules, and these single-stranded DNA molecules inhibit mRNA needed to produce huntingtin. Symptoms of Huntington disease can start at any time, but usually develop between 30 to 50 years of age. The likelihood and age when symptoms start are linked to the number of CAG base sequence repeats in the gene for Huntington's disease. This phenomenon is actually known as anticipation for those that want an extra fact. genetic anticipation. However, recent studies have suggested that epigenetics may affect the age when symptoms first start. Damage to the myelin sheath of neurons can cause muscular paralysis. Explain how. So, just a quick recap on neuroanatomy. So, you've got a nerve here, and you've got your myelin here. The myelin acts as an insulator. So, what happens is rather than the impulse traveling the entire thing, which takes ages, it jumps like this, which is much faster. That's called saltatory conduction. And it jumps from nodes of Ranvier. So, nodes of Ranvier are just bits where you have no myelin, they're just nerve. So, that's what we're going to talk about. So, we've got reduced saltatory conduction. So, depolarization has to travel whole nerve length. So, that this means that impulses are slower or stop. Therefore, neuromuscular junctions, because it's asking us about muscular paralysis. So obviously, therefore, neuromuscular junctions are stimulated less. You need to mention the impact it has on the muscle itself. You can't just say, oh, muscles can't work. It has to be either the neuromuscular junction, which is the nervous connection of muscles to the nervous system, or the sarcolemma. So you have to talk about the nervous system aspect of it. Okay, so sometimes Guillain-Barré syndrome can cause heart rate irregularities, suggest and explain why. You need to think about how the heart rate is controlled. So the heart rate is controlled by parasympathetic and sympathetic neurons. So, okay, just as a recap, you've got receptors like chemoreceptors, baroreceptors, whatever. They pick up stimulus and they send a signal to the medulla. The medulla then sends signals via the parasympathetic nervous system or the sympathetic nervous system, depending on which one, to the SAN in the heart and alter its firing rate. So Guillain-Barré syndrome is where you've got damage to myelin sheath and neurons. So if you've got damage, then what you're going to have, what we've just mentioned here is that the conduction's slower. So the conduction from the medulla across the PNS or SNS to the to the sinoatrial node is going to be reduced. So that's what I'm going to say. So there's slower or fewer impulses along. I'm going to write PNS for short and SNS, but you write the whole thing. PNS or SNS neurons from the medulla to the SAN. As a reminder, the heart is myogenic, so it beats on its own, but the medulla and the parasympathetic nervous system and sympathetic nervous system influence how fast it fires. The first successful drug trial to reduce concentrations of huntingtin in the brain used single-stranded DNA molecules. Suggest and explain how this drug could cause a reduction in the concentration of the protein huntingtin. So this one's testing. So just as a quick reminder, it says where is it? It inhibits mRNA needed to produce huntingtin. So this is testing your ability to apply this to protein synthesis. So if you don't know your protein synthesis, please, for the love of God, go and look at it. So you've got your mRNA here, right? You've got your transcription where, all of that stuff happens. You've got mRNA, and the mRNA goes to ribosomes in which it's translated, and you get Huntington produced. So something's stopping that from happening, and something's stopping that from happening. What's actually happening is this single-stranded DNA here binds to it like that because it's complementary because of the complementary base pairs, and it stops it from being translated. So the single-stranded DNA is complementary. You obviously write in full. I'm writing it in short because I'm keeping, track of time. To the mRNA for Huntington. Therefore, it binds and it stops translation. If you have no translation, no Huntington. The whole of this sort of genetics topic is testing your ability to apply transcription and translation. So please go back and learn it properly if you don't understand it. Okay, scientists from the first successful drug trial to reduce concentrations of Huntington reported that the drug is not a cure for Huntington's disease. Suggest two reasons why the drug should not be considered a cure. Do not include repeats of the drug trial in your answer. So when you get questions like these, you should sort of assume that scientists have followed good protocol. If we look at the data that was given to us in the question, it's tested 46 patients. The concentration of Huntington was reduced. It was only for 4 months. However, we know nothing about the symptoms. Like, it hasn't mentioned anything about symptoms. Um, it only says it's been reduced, so they're still probably making Huntington proteins. 4 months is a very short amount of time, and 46 is a very small sample size. So that's a lot of things you could talk about. Also, like, brain damage may continue as a result of other things, not just only Huntington. So I'm going to say small sample size and only 4 months treatment. Because, for example, for the small sample size thing, if I got a room, and I use this example all the time, If I had a room full of 10 people and I gave each one of them chocolate and every single one of them for some reason had explosive diarrhea, that's 100% of the people I tested having explosive diarrhea when I ate chocolate. That doesn't mean that if I give a million people that, all 1 million of them are going to get explosive diarrhea because, we both know that just doesn't happen. The reason for that is because the sample size I had is really small, so chance plays a bigger role. And for those that do get explosive diarrhea when eating chocolate, I'm very sorry for using you guys as an example. Now, in regards to the 4-month trial period, well, 4 months is really short. Ideally, when we want these things, we want really long-term stuff because there might be side effects that can come along or it might stop working after a while. And in regards to the Huntington protein itself, it's only been reduced, it's not stopped, so there's still some being made. Also, it might just be temporary. It might be that you have to keep giving them the drug to keep having that effect working, and it might be like continuously. And as well as that, it leads to brain damage, as mentioned. So the brain's already been damaged and it might not regenerate. Okay, so it says suggest two reasons why people have the drug injected into the cerebrospinal fluid rather than taking a pill concentrating containing the drug, sorry. So if you look at the data, it says that the cerebrospinal fluid bathes the brain and spinal cord, and the problem itself is in the brain. So rather than taking a tablet and relying on it to go through your entire gastrointestinal tract and absorbed into the blood, the drug reaches the brain directly. Now, that's obviously one thing. What happens when we take tablets? Well, we've got stomach acid that will absolutely batter it, so we're not destroyed by acid. Obviously, we have ways around it, or else we wouldn't be able to take any meds, at all by mouth. But in this case, that's what they're looking for. So now it says suggest and explain one way epigenetics may affect the age at which symptoms of Huntington's disease may start. So epigenetics, when you're thinking of epigenetics, there's two main things you want to be thinking of: methylation and acetylation of histones. All right. Some increased methylation leads to less expression. Decreased methylation leads to increased expression. That's because methylation blocks, it makes it harder for RNA polymerase to do its job, basically to produce mRNA. The opposite happens for acetylation. So if you have increased acetylation, you've got more expression because it causes the histones to sort of the chromosomes to unfold and coil in such a way that it's easier for RNA transcriptase to do its work, and decreased acetylation leads to what is it? Decreased expression. So you can pick either one of these and talk about it. So, for example, I'm going to say decreased methylation leads to transcription of mRNA for Huntington. Thank you for watching, and please share if you found this video helpful.